The Marine Ecology Lab at SERC has been studying the prevalence of a parasite and subsequent disease called perkensis, also known as dermo, in oysters in the Chesapeake Bay. Dermo affects blood cells of oysters and is not harmful to humans. However, heavily infected oysters usually don't grow at the same rate as healthy oysters, and their ability to reproduce is affected. Dermo disease has been present in the Chesapeake Bay for over 20 years and has been a large contributor to oyster mortality.
So this project involves dissecting and analyzing over 700 oysters. So this year we decided to bring in some extra hands, and we called citizen science members and volunteers to help us with our analysis.
We've had citizen scientists really involved in all aspects of the project, from getting very muddy, pulling cages of oysters up onto the dock, to dissecting out the digestive systems, to recording data, helping make graphs, really just about any part of the research that takes multiple hands to do.
We start by taking oyster cages out of the field and bringing them up to the lab, where the oysters are washed and shucked, and we remove 2 tissues to look for dermo disease in oysters. The first tissue we pull is mantle tissue, which is responsible for helping an oyster build its shell. And the second tissue we pull is anal tissue, because it's the last stop in the oyster's digestive system. Both of these tissues are useful because dermo is a waterborne disease, and it will frequently appear in anal tissue, and in low infection intensities, mantle tissue adds extra tissue to look at for diseased cells. We'll sit our tissues in this media for about a week at close to room temperature, and then tissues are put on microscope slides with some Lugol's iodine, which will stain the diseased cells a dark blackish blue that's easily read under the microscope. There are several different ways dermo cells can be scored across a range of scales. Our lab in particular uses a Mackin scale, which ranks disease infections in tissues on a 0 to 5 ranking system.
Lower scores indicate fewer dermo cells in a standardized field. A higher count of dermo cells in a field will yield a higher score rating of the disease. A score higher than 4 indicates that dermo is detrimental and possibly lethal to the oyster.
The Marine Ecology Lab wants to extend a huge thank you to all citizen scientists who helped during this project.
We really wouldn't have been able to get the project done without all the help from the great people we had.